Hello, my name is Mallory Bao, and this is my presentation on types of cell competition. Cell competition occurs primarily for two reasons. One is the elimination of a cell or cells, or two, proliferation of a cell type. Elimination of cells generally occurs for two reasons. One, because a cell or cells are damaged. This is primarily a mature cell issue. Or two, because during development, cells are identified as not being fit enough, so they are eliminated in favor of cells that possess more fit qualities. Proliferation of a cell type occurs generally during development, as cells are necessary for growth and development is proceeding at an increased rate. But it may occur at other times too, say in the case of a wound. In the article that I will present, the cell competition falls into this area, tissue sizing, or maintenance of homeostasis. While reading the review article, I found the section on mechanostretching to be particularly interesting. After much searching, I finally found this article, Mechanical Stretching Triggers Rapid Epithelial Cell Division Through Piezo-1, which details a mechanical method of cell competition, specifically through the piezo-1 receptor. In the article, the question being addressed by the researchers was, how is it possible for an epithelial cell to regulate its cell division and maintain an optimal cell density, given that there are so many types of epithelial cells in the human body covering all sorts of openings at all sorts of various depths and being of all sorts of densities? They were interested in determining how the number of dying epithelial cells always matched those of the epithelial cells that were being regenerated. The authors were interested in researching this topic for the listed reason. It is unclear how epithelial cell division is controlled to balance cell death at the steady state. This topic is important because our understanding of this mechanism enables us to understand cell regeneration and growth, either during growing times or after a wound when cells are replacing themselves, or as a possible mechanism for tumor suppression. In this research study, the researchers were using Madden-Darby canine kidney cells. And they were cultured with a high glucose FBS, penicillin, streptomycin, and everything was held at 37 degrees Celsius. And they performed 6 different tests or assays as they were conducting their research. And I will go over all of those different tests that they went through and the results and what the impact of those results are. So the first test that the researchers performed was to determine at what point do cells reach a steady state. And this is known as their density. To perform this test, the researchers immunostained the cells for the phosphohistone H3 and watched them divide. They determined that the steady state or rate at which cell division slowed wasn't until day 5. And that is seen here, and you can see as it proceeds, as those phosphohistone H3 tagged cells, day 5 is when cell growth slows. It should be noted, however, that cell growth does not stop as seen in days 6 and 7, it just slows considerably. The density of the cells around the 5th day of growth was approximately 11 per 1,000 micrometers squared. And that can be seen down here in graph B as it raises right here around day 5 and then plateaus. This video from their research shows that rate of cell division, which is pretty amazing to watch. And you can see the maintenance of that homeostasis. We are getting cells where we need them. Based on observations of cellular division, continuing to occur at low density subregions after the monolayer had reached, so the top layer had reached a steady state, led the researchers to test if cell stretch due to low density was activating the epithelial cell division. So to test this, the researchers experimentally stretched cells in 2 ways, both by wounding them or by using what is shown here in figure A, a stretch plate, which is has been 3D printed. The researchers found that a 5-fold increase in mitosis occurred within 1 hour of stretching or wounding, and that a return to homeostatic levels resulted quickly within 4 hours using the stretch method. The wounding done by a scratch method also produced the same results. This video shows the scratch test and the resulting cell regeneration back to density. You can see the empty scratch, and then the cells very quickly, this is a time-lapse video, do regenerate themselves back to their original state. So during these tests, researchers also used, next the researchers were interested in how this stretch-induced mitosis was regulated. In order to determine which protein was causing the regulation, they inhibited a variety of other known mechanisms. Gladiolum, a generic stretch channel inhibitor, was used, as well as roscovitin, a CDK1 inhibitor, and then a knockdown of piezo-1, either using short interfering RNA or short hairpin RNA, were all tested. And it was found that piezo-1 knockdown reduced the mitotic rate back down to within 7%. It reduced the piezo receptors down to 7% of wild type levels. So there was a tremendous loss when they were knocked out. As it should be seen, they were physically removed via those hairpin RNA or interfering RNAs. What that did was it ended up blocking, and you can see this over here, it ended up blocking the ability of the cell to go through mitosis. So not stopping here, however, the researchers next added a GFP tagged piezo-1 back into the cells that had been targeted by those, so the same cells that were targeted by short hairpin RNAs and the short interfering RNAs, and it found that those rates of meiotic division were rescued, and that can be seen here in this blue bar that returns to normal rates, as well as in this Western blot analysis, you have the actual protein that can be found. Again, the mechanoreceptor is present. During these tests, the researchers also used a CRISPR technique to knock down piezo-1 mosaically. So they found that doing this caused a 50% reduction, right here, in mRNA production of the piezo-1, and a 5-fold reduction in the phosphohistone H3 cells. They also tried this using phosphoactivated morpholino techniques aimed again at the piezo-1 receptor, and they also found that there was a tremendous reduction in the epithelial cell proliferation. That can be seen here. These tests are both with the CRISPR method. In conclusion, at this point, the researchers were able to identify that epithelial cells at the steady state become stretched before they divide. That stretch is sufficient to activate rapid cell division, and cell division requires the stretch-activated channel, piezo-1. And this is a model of that piezo-1 receptor. And it is a transmembrane receptor. So following this conclusion, the researchers were interested in determining, well, where in the cell cycle does piezo-1 receptor need to be stretched in order to induce cell proliferation? So to test this, the researchers tested if stretch simulated cyclin B, which as we know accumulates in the cytoplasm prior to the cell entering G2. It is a checkpoint that has to be cleared. So they measured cyclin B, which degrades pretty quickly. As soon as the amount has been discovered, it will then proceed to degrade. And they tested it as the cell moved into mitosis using roscovitin, which is that CDK1 inhibitor. And they found that stretch does increase levels of cyclin B. They then ran the test in a converse manner, and they inhibited piezo-1 with gadolinium, or an alpha-amothin blocked cyclin B production, an amothin and they found that cyclin B production was also inhibited when the stretch was prevented. So the results of this are present here in graph A, the control time after stretch and the amount of cyclin B. You can see it's normal. When there is no stretch, that cyclin B does not build up in the cytoplasm, so G2 would not begin. The alpha and amytin and the gadolinium, you can see both here, block the ability of the cell to build up the cyclin B and be able to enter into G2 phase and proceed to mitosis. To confirm their results, the researchers knew that piezo-1, the transmembrane protein, also relays calcium when mechanically stretched. So therefore, they assumed that inhibition of calcium should also inhibit the cell cycle. And this is shown here in figures B and C, that with stretch, there would be proliferation, and without stretch, there wouldn't be. The following tests, all three here, were run, and they were run to test if cyclin B would be produced and also if these HP3 cells would be produced in the end. And they found on both of those, when you inhibit that piezo-1 receptor, and when you inhibit calcium, you do not get cells to move into the next phase. So the researchers inhibited calcium movement and found that both of those productions were stopped. Next, the researchers were interested in what induced proliferation in response to calcium influx. They noticed that a single spark of calcium preceded mitosis entry. So what they did was performed a phospho-ERK 1,2 immunoblot, and it was found that stretch-induced calcium activity does activate the pathway that is needed with the kinase to within 5 minutes. So this ERK pathway within 5 minutes of receiving calcium was triggered and it got started. And this diagram here in E shows in a nice flow chart how stretch does induce mitosis, and it is through this sequence of events. So following all of that, the researchers, one comes to understand that piezo-1 controls both cell division and cell death in order to maintain homeostasis and regulate epithelial cell numbers. The graphs here in A and B show that it does this through either a stretching mechanism or an overcrowding mechanism. And it does all of that using the ion calcium. Interestingly, when the researchers were doing this, they thought, that's weird. One ion and one receptor have both a negative and a positive feedback. This is known as a paradoxal feedback loop. And it is similar to the one for interleukin-2, the way that it controls the homeostasis of T cells by its amount, either allowing their division or cell proliferation or their death, depending on how much IL-2 is present. And finally, the researchers wanted to understand how is it that piezo-1 is able to differentiate between crowding and stretching. And they found that it was not only those cells that were primed to enter, that it was only those cells that were primed to enter G2 that were affected. So cells that were still stuck in G0 or cells that hadn't cleared S phase weren't impacted by either stretching or crowding. Only those cells that are primed and ready to enter G2 would have the impact on the piezo-1 receptor. They are dissatisfied, the researchers, with their understanding of this mechanism, how the piezo-1 receptor can differentiate between being crowded or being stretched. And this is an area of further research that they would like to explore. And they realize that likely in here there is a connection with piezo-1 and YAP, TAZ, and the beta-catenin pathways. So based on this new understanding, it is thought that because piezo-1 controls two processes, its downregulation should slow both processes, division and death. If this were the case, this means that it could be a possible target for